Welcome students, and today's topic is reproduction in bacteria and certain more examples in kingdom Monera. Anyways, that as you people are going to give the NEET exam in 2022, the thing is, if you are going to get guided rightly, if you are focused for your target, and I said this thing in the last class also, that dream with open eyes, then definitely you are able to make it possible. To get a medical seat is not at all a difficult thing. The most difficult is to follow that path which lead to success. So be clear about one thing, that get focused, just focus to your target, just dreaming about your target all the time, and execute the planning. If you are a successful executioner of your plan, definitely you are going to achieve the goal, the medical seat in NEET 2022 or the highest score in the subject. Definitely you can make it possible. Anyways, the topic that we discussed in the last class, for that we have already uploaded the DPPs, and I am still saying that if you are following the classes, you need to go for Yupp Master app if you are watching on some other platform, whether it is YouTube Live or if it is AP FiberNet, Egg FiberNet, or on your smart TVs. It is better to download the Yupp Master app and attempt that daily practice papers as well as weekend papers which are available on the application. Welcome to Yupp Master, and here we are going to start our topic for today, that is reproduction in bacteria. Remember one thing, my students, that reproduction in bacteria is asexual. Means we are not at all discussing the type of things which we can say that bacteria reproduces sexually. So what type of thing we can mention over here? It's quite simple. First of all, when we are defining about reproduction, you should remember one thing, that in biology only, means the biology alone is a subject where we say that divide, multiply or add, these all are increasing the number of cells. Obviously, here the meanings means same for us. This novelty is seen in biology only, nowhere else. Okay, means a cell divides, a cell multiply, a cell adds the number, so it is the same. The number of cells are increasing. So the fission word actually means division. The fission word means actually division. So what type of division they are showing? Because this is asexual, in that cases, if it is asexual, remember one thing, that asexual reproduction is mitotic. Asexual reproduction is mitotic. Means as the parent cell, the same is the daughter cell. Okay. So if bacteria are reproducing asexually, in that cases, I am again getting one single word for the bacteria. Previously, means in the previous topics of the kingdom Monera, about the previous character of kingdom Monera, we have discussed certain things which can be mentioned in the single word. What are those things? See here, just a recall of the things that we have studied earlier. For example, the first thing I told you was about occurrence. Means where these examples are found, and here we are saying cosmopolitan, everywhere, here, there, everywhere. Okay. Number second character that I can write here, is about their morphology. The morphology of bacteria is diverse. One word only. Okay. Next comes about structure. For structure, I told you their structure is simple. Their structure is simple. Next character or the next feature that we can write. Okay. The next feature that I can write here is nutrition. Mostly heterotrophs. Again, about their behavior, complex. One more feature. Out of these, now I am adding one more feature here, and that is what? Reproduction. And in them, reproduction is asexual, and that too by binary fission. So for all these characters, we can use the single word, right? So this is how you can retain the things that we have studied so far about the characters of kingdom Monera, the monerans, the prokaryotes, the bacteria. Clear? So here I am saying that bacteria reproduce mainly by fission. So here we say if asexual reproduction is common in bacteria and that is taking place by binary fission, then the most common method that we are observing in bacteria is binary fission. Binary fission is dividing into 2. Binary means 2, and thereafter multiple of 2 will be formed. Okay. So in that cases, here I am getting that like any asexual method, fission is rapid. Like any asexual method, fission is rapid. Okay. But the situation is not same when we are going to study it in a laboratory. We will discuss it later on. As I said, the fission is binary. Okay, means 2 multiple of 2 cells. So here I am telling you about the very special feature of this thing, or multiplication in bacteria. What is that? You can consider if at a time 2 cells are getting formed. At a time, if 2 cells are getting formed, then I can say one thing very clearly, that each cell is dividing into 2. This is one cell, getting split into 2, forming the 2 cells then. So these are 2 daughter cells, fine. Okay. So if it is so, in that cases I will say the number of cells formed, will be equal to 2 to the power n. The number of cells formed will be 2 to the power n, fine, where n is number of generations and can be obtained by total time given to me, means to the cell which is dividing, upon the generation time, that in how much time a cell divides. You can write in abbreviated form as T and G. Okay. Let's see if all cells are dividing at once. Question first type, that I am saying that this is a glass whose entire volume is V, and they are saying that this is half filled. This is V by 2, and it took 18 minutes to get filled into half. Okay. These are all bacterial colonies occupying half volume of the glass, right, or is this vessel. Fine. If I am saying this, if each cell divides in 1 minute, how much time will it take to fill the vessel? Time will it take to fill the vessel? How much? Just 1 more minute and they will get doubled, because each cell is requiring 1 minute. So I will just answer it what? 1 more minute in total. 1 more minute, that is 19 minutes in total, and the glass will be full. This is one of the type of question, logical based. So numericals are also there in biology. We need to be very clear about them. We need to understand the fact behind them, and we can answer them very easily, without any doubt, without any problem. Another type of question. A bacterial colony with 10 raised to power 15 cells. If each cell divides in 35 minutes, then total cell or total size of colony after 175 minutes will be. Now what is given to me and what I can find it out, they see this 35 minutes is the generation time, means small g, this one. 175 means total time, small t. So here I will say I can determine the number of generation. How? 175 divided by 35. This comes out to be 5. Okay, this will be the number of generation. So I am considering what? Initially the number of cells are 10 raised to power 15. Then what will be the number of cells after time t? So it will be 10 raised to power 15 into 2 to the power n. What is 2 to the power n here? 5. So answer must be 32 into 10 raised to power 15 cells. That's it, answer. This is my answer. Okay, so fast we are going. So this is what I said, that when binary fission is taking place, it is much faster. We are considering that all cells are dividing, and this is how the number of cells get increased in very less time. Fine. But in the laboratory the situations are different. How the situation in a laboratory get different? We'll get on to this, but first of all we need to study the steps that how the binary fission is taking place, right? Coming to that one. So here we say that before a cell divide, it is necessary for the cell to undergo DNA replication, or you can say the genetic material is getting replicated or duplicated, right? The genetic material first get duplicated. Fine. Here I am saying that this particular substance is representing. This substance is representing the viral genome. Although not given in the diagram because it is not explained at your status, but there is one structure that is called as mesosome. Now what does it mean? Mesosome means the plasma membrane foldings, which we were studying at the time of respiration as well, if you can recall about respiration in bacteria. At that time I told you the word, the foldings of plasma membrane where aerobic respiration is getting completed in bacteria, mesosomes. So those mesosomes are considerable here. Here I am telling you the things about mesosomes. See, this is the plasma membrane and we are observing that it is having certain folds. Now we call this one as peripheral mesosome, and this one as septal mesosome. This structure is septal mesosome. So mesosome are of two kinds, septal and peripheral. Okay, one is septal and the other one is peripheral. Fine. So these are the things that we are discussing over here, and the mesosome is going to participate in cell division. How it participated? See, in the coming stages we are saying the DNA is getting replicated. Fine. At this time, this septal mesosome who is involved in division of cell also, what happened? The mesosome also splits. See, one portion here, other portion here, and each of the portion of mesosome is holding the DNA with it. Each portion is holding DNA with it. So this is how the condition happened. See this, right? So if it is so, in that cases we are mentioning that they are holding their own DNA. If they are holding their own DNA, then we are saying that what will happen next? Each mesosome is having its own DNA right now, one copy to each. In the subsequent stages, these mesosomes are letting the DNA particles to move towards different ends. See, this is what happened. Okay, the mesosome I am going to make again. Here it is. Now these mesosomes have moved apart. Now actually this is the time when this plasma membrane will grow towards inside and letting the cell wall also to penetrate inside. Means this is the cross wall formation. See, this is how the things are going on. This is cross wall formation, and thereafter we are observing the two cells are getting divided and we got two daughter cells which are alike. Fine. So this is how we can study about the mechanism, the steps taken place during binary fission. Okay, in words. In words I'll write. Steps in binary fission. What is mandatory? Replication of DNA, or you can say duplication, copying of DNA. Second, splitting of mesosomes. Third thing, cross wall formation. Fourth, simple, just simple. I got two daughter cells. Okay, so these are the steps that we are observing during binary fission. Now, as I told you that this kind of binary fission is not going to be the same in laboratory condition. Outside the lab or in the natural condition, means around us right now, they are dividing without any hustle, without any problem. They are getting divided, they are forming the new cells. Okay, but how we are discussing that the situation become different in the laboratory, here it comes. Here is the situation that we need to tell about or we need to discuss. That is, remember the steps. Okay, and now what is happening? See, as the cell is going to increase in size, it will divide. Okay, first the cell will increase in size, means grows in size, then multiply or divide or undergo fission. Okay, so this is number of cells. And this is plotted against time. Fine. So initially the things are like this. The initial situation was like this. I called it as A. Means where I am talking about these things, this is a petri dish. Okay, this is a petri dish where we are considering all the things, means the bacterial cell here, these are bacterial colonies. These are bacterial colonies here. Right? So what is it? This is the culture medium. And what are these? Are bacterial colonies. These will be bacterial colonies then. Fine. So these are the things that we need to discuss about. After a while, when the cells are getting multiplied or they are increasing in number, replicating or dividing cells. right? Replicating or dividing cells. Now what is happening with them right now? We are saying that soon they are going to occupy most of the vessel. They are going to occupy most of the vessel now. So the earlier formed colony and newly formed colony, both are here. These are earlier colonies. They have occupied a lot of space. Now the new colonies are formed as well. Means the space is getting lesser with the time. The space is getting lesser with the time. Okay. So this is how we are saying that colonies are becoming densely colonized. Okay, they are getting densely colonized. Newly formed colonies also I am telling you. Here are the newly formed colonies. Here. But problem is that as the colonies are increasing, they are occupying more spaces. What is happening right now? In that cases, what will happen? As the number of cells increase, the space decrease. The available space is getting lesser. Replicating or dividing cells plus fall in or decrease in available space. So now if the available space is getting decreased, you cannot add further nutrient to this. Clear? You cannot add further nutrient to the growing colonies. Then how they are going to survive? They don't get much of the food. And number two, if they are too much in number, means if the cell types are too much in number, they will get attracted. They are attracting their predators. Yeah, the same happened. Clear? So the number increased very fastly. Thereafter it becomes almost stable, means unchanging. Where they were increasing very fast, I said it as phase B. Now they are getting almost unchanged in number. C means as many as new formed, that many number died. Now the number of dying cells get increased. Why? Because the dead cell in the same space created toxicity, and now the number is finally declining. So the natural condition for binary fission of bacteria is different, and in the culture medium it's different. We are getting these phases in the culture medium. I can say that what are the names of these stages or how you can represent. You can say here in the A the growth was slow. In the B the growth become exponential, means you can say too fast. In the stage C, what happened? I am saying it is steady phase. And in the case of D, it is declining. Okay. So 1, 2, 3, 4 stages are there. The question is not about the stage A, B or C. The question is asked about the stage D, why it is happening. Clear? What is the reason? Declining. Probable reason of declining I need to tell. I need to mention that why the number of cells are decreasing or why the graph is going down, why the curve is becoming down. Probable reasons. Reason number 1. The reason number 1 as we are discussing here is, the first reason is quite obvious that we can mention here, that the probable reason for decrease or decline in the number is unavailability of space. Means I cannot add further nutrient to this medium. Number 2, the dying cells are increasing toxicity. Number 3, the other condition that I can mention over here is invasion of predator. And the predator of bacteria. Who is the predator of bacteria? Well known, it is bacteriophage, the virus which feeds on bacteria. That may be the reason that this situation is going to decline. Fine. So remember how the graph of bacteria or bacterial cell culture is there in laboratory medium. Okay, this is what we learned about. Why the phase is showing declination, or we are saying that why the number of cells are decreasing. So here are the possible reasons. Fine. This is about the binary fission. Moving to the next thing, but remember, binary fission is the thing which is observed during favorable condition. Means the cells reproduces asexually by binary fission during favorable conditions. Bacteria reproduces asexually by binary fission during favorable conditions. So if it is so, in that cases I have good enough reasons for this. Oh, it means now the situation, if unfavorable, what the cell is going to do? Because they have to reproduce asexually, and asexual reproduction is common in favorable condition. So many bacteria are exhibiting a mechanism to escape the unfavorable situations. Means when the nutrient is not available, when the temperature is not favorable, when the moisture or any other conditions are not favorable, at that time the bacteria is trying to escape itself. Clear? By forming certain thick walled structures. We call them as spores. Clear? We used to call them as spores in that cases. Fine. So here it is the thing that I am going to tell you here, and here it is. Here it comes. That what are the things that what they are going to do, particularly not all the type of bacteria, remember this. During unfavorable season or unfavorable conditions, right? During unfavorable conditions, what they are going to do, particularly which type of bacteria? Rod-shaped bacteria. Tell me, what is the name of rod-shaped bacteria? Rod-shaped bacteria are called as bacilli. Okay, let me tell you the example. Lactobacillus, which is present in milk. Okay, and about the unfavorable situation as well. The milk is sent for pasteurization. Usually below 100 degrees Celsius. There are different methods of pasteurization. One is called as long time less temperature, or it may be like less time high temperature, or ultra heat treatment. Ultra heat treatment, less time high temperature, sorry, LTHT. Long time less time high temperature, and the other one is less time then high temperature, and when long time it is less temperature. Okay, so that is the thing we are saying. Here it is long time, this is less temperature, this one less time, then this is high temperature. There are different methods of pasteurization. Pasteurization is going to do what? Pasteurization is going to create unfavorable situation for the rod-shaped bacteria. So it realized, oh, the situation is not good for us. It is better to avoid multiplication right now. So pasteurization acts as bacteriostatic method. Act as bacteriostatic method. not bactericidal. Remember, it is not bactericidal. Killing the bacteria, bactericidal. Right? Killing the bacteria is bactericidal, but avoiding the multiplication is bacteriostatic. It is not bactericidal. Remember this. So I am saying bacteriostatic method, as the rod shaped bacteria formed escapes by forming the spore like structure, which are here named as endospores. We will call it as endospores. You remember the word endo, only spores. The cell is getting surrounded by a thick coat which is resistant to temperature type of things, which are provided during pasteurization. So the cell is escaping pasteurization. They are not at all getting killed by pasteurization or that temperature. They just become inactive. And whenever, say for example, if you bought the milk and you forgot to boil it, if you forgot to keep it in the refrigerator, the milk will become sour or it will form curd. Why? Because lactobacillus become activated. Okay, in the dairy plant, they may have pasteurized the milk. The milkman may have boiled the milk, but whenever the time is becoming more, means the delivery time from the dairy plant to your home, after that it is recommended that you should boil the milk or keep it in the refrigerator for safety purpose, or else the milk will become sour. So we are saying why why the milk is getting sour, because the bacterial cells are coming out from their endospores and they are multiplying. When they start multiplying, they will make the milk sour. So that's why we are saying pasteurized milk can be converted into curd. Sterilization, if done, that milk cannot be converted into curd. That cannot be made sour. Sterilization is different. It is bactericidal. Pasteurization is bacteriostatic. Okay, be clear about these things. So endospores can escape pasteurization because they are thermal resistant. Not at all affected by pasteurization. The cell will survive by a thick wall around them. Okay, so this is during unfavorable condition. True sexual reproduction is not found in bacteria. So here characters are mentioned. Now what are these actually? Meiosis, fusion of gametes, gene transfer or recombination. These are features of sexual reproduction. If I am going to compare it with the bacteria, I have observed that bacteria is not showing meiosis, bacteria is not showing fusion of gametes, bacteria is just showing gene transfer or recombination. Let me tell you this thing, fusion of gametes means syngamy or fertilization. Okay, so that is not observed in bacteria. So one more character I can add, and that is what? The other character that I can add here is alteration of generation. Means there is no alteration of generation, there is no alteration between haploid and diploid phases. There is no alteration between haploid and diploid phases too. Not present. So only one of the feature of sexual reproduction is present in bacteria, and that is what? The gene transfer. Let's compare with the actual type of sexual reproduction in the eukaryotic cell, therefore, that how the eukaryotic cells are getting different. This one, this one, this one, these three things, as well as alteration of generation. These all are present in eukaryotic organisms, but not present in bacteria. So here is one only comparison. So I can say the sexual reproduction in bacteria is comparable to, or the sort of sexual reproduction in bacteria is comparable to eukaryotic organisms only on the basis of gene transfer or gene recombination. Okay, the sort of sexual reproduction, I am not calling it a sexual reproduction, sort of, so called sort of sexual reproduction in bacteria is comparable to eukaryotic cells due to gene transfer or recombination. Okay, it is due to gene transfer or recombination type of factor. Keep these things in your mind. Okay, the sort of sexual reproduction in bacteria is comparable to eukaryotic cell or due to gene transfer or recombination. This is what we have learned. Fine. Okay, so what actually happened in bacteria? Why we are saying that it is not showing meiosis or any fusion of gametes type of things? The thing is that is not at all possible in them. Number secondly, they are showing gene transfer, but how? Actually, the gene transfer that is taking place in the bacteria is involving two strains of bacteria. One is the donor, other is the recipient. Just like in sexual reproduction, two individuals, one is the male, the donor, and the recipient, the female, participate. The same is in the bacterial cell also. Okay, but remember, in eukaryotic organism, the participation of male and female individuals in sexual reproduction is going to give offspring. In bacteria, the participation of the plus means the donor, minus means the recipient strain, means the male and female respectively, is going to change the gene composition of the recipient. No new number of individuals added. Clear? The population size remains the same. Only thing getting changed is gene expression, that too in recipient, not in the donor. Remember, means here the sexual reproduction is not increasing the number of cells, it is not increasing the population, it is increasing only and only what? It is increasing only the gene combinations, that's it, in the population. So here is the thing. So that is why the sort of sexual reproduction in bacteria is a primitive kind of gene transfer. If it is a primitive kind of gene transfer, then what does it mean? We have three different experiments to confirm these things. Okay, what are those? One is conjugation, which is performed by Lederberg and Tatum. Okay, these are the persons who are considered to be the discoverers, and they studied on Escherichia coli for this. They have taken Escherichia coli, the bacteria which is present in our colon, was taken by them. Number two, the donor and the recipient strain. As taken in conjugation are in same medium. That too in physical contact, they are in touch with each other, so that the gene can be transferred from the donor to the recipient. That kind of thing they are telling. Okay. Number second, means I am saying they were in contact, they were in the same medium. Fine. The second type of experiment conducted by Frederick Griffith, it is named as transformation. We can call it as transformation effect also. Okay. So this was by Frederick Griffith, and he studied on the bacteria that causes pneumonia, Streptococcus pneumoniae, or you can use the word pneumococcus as well. Now here the plus and minus strain means the donor and the recipient, the male or the female, like this, are going to be what? They are present in same medium. Okay, they are present in same medium, but not in contact. Oh, it is like the donor shared the genetic material in the medium and the recipient absorbed it from the medium. This is the mechanism in which transformation is studied. Okay. So, transformation is something by taking the genetic material from the medium, not through the contact. Okay. So, fine, at least they are in same medium, fine. The third type of experiment was done by Zinder and Lederberg. Zinder and Lederberg performed the third kind of experiment. And what is the third kind of experiment? This is not different one. The plus and minus strains are not in contact. Actually, they are not at all in the same medium. Then, how the transfer of gene is possible? It is by the agent. Which agent? The bacteriophage, which invaded the donor strain first, took up the DNA of the donor and get transported and taking it simply to the recipient. This is how the gene from the donor get transferred to the recipient cell. So, means in the third case, in transduction, there is a kind of agent involved on what they were studying. A Salmonella typhimurium that causes food poisoning. So, here as I am saying, the plus and minus strain are invaded by bacteriophage, means a kind of virus which feeds on bacteria, which acted as agent for gene transfer. That acted as agent for gene transfer. So, that type of character we are telling here, that the conjugation, transformation and transduction are these three types. These are three different types of primitive kind of gene transfer in bacteria studied so far, and they are different in the terms that how the gene is getting transferred. Remember, in bacteria during so called sexual reproduction, there is no new individual born. There is no addition of the number of individuals to the population. Let's compare therefore in the brief that how the sexual reproduction in bacteria, sort of sexual reproduction in bacteria and the true sexual reproduction in eukaryotic cells are different. Eukaryotic cells and bacterial cells. Union of gametes form zygote. Whereas if bacteria is considered, in that cases I will say the recombination of genes from where? From donor and recipient as merozygote. Now the name is different. Remember, this is not a zygote. This is called as merozygote. Okay. Zygote into a new individual. Zygote is going to be a new individual soon. Zygote is going to develop into new individual in the population. And here we say no new individual is added. To the population. Then what's the last? The last thing is very simple that we can mention over here, and that is that if we are considering these type of example, then I can say This type of sexual reproduction is called as amphimixis. This is called as amphimixis, whereas in bacteria we used to name it as meromixis. The two terms are obviously different. The whole mechanism is different. The bacterial cells are not undergoing true kind of sexual reproduction. Remember this, memorize this, retain these things as long as possible. Okay, hopefully they are not going to ask these terms like meromixis, but you should know that how the things are different. Okay, there is a more in-depth study of this type of meromixis or formation of merozygote as well. Okay, now that much is not told in the classes. The reason is for many years such questions are not appearing in the exams. Okay, here is the sexual reproduction, sort of sexual reproduction bacteria that we have studied. Now, so this is towards the conclusion of bacteria or the kingdom Monera. One example which is still important for us is Mycoplasma. Now the Mycoplasma is something different. Means something different in what term? The brief things that they have mentioned in the NCERT are here. But we need to know a little bit more about these monerans or these Mycoplasma in particular. So what about them? They completely lack cell wall. What does it mean by saying completely lack cell wall? So it is simply, I can say, they completely lack cell wall. Why? Means whatever you are going to feed them, they will never attain a cell wall. Okay, they will never attain a cell wall, no chance. Okay. Secondly, they are the smallest living cells. They are smallest. I am going to derive things out of this. Next, and can survive without oxygen. Okay, very nice. It means they are actually aerobic, but if oxygen is not available, they can start respiring anaerobically, and such type of examples are called as facultative anaerobic respiration for time being. Many Mycoplasma are pathogenic in animals and plants. This is true. Many pathogenic examples are there that are causing disease in animals as well as in plants. Okay, anyways, this is the intro. See how the relevant things are derived. First, I want to know about this. When they say completely lack, now is there any bacteria which is not lacking the cell wall for complete phases? Yeah. Or the bacteria which can lose the cell wall and then again can achieve the cell wall around it? Yes, it's possible. Examples are there. Okay, let's discuss about them in the detail, how those Mycoplasma characters are different. So remember, Mycoplasma are also considered as a connecting link between virus and bacteria. There are so many terms for Mycoplasma. We used to call them as PPLOs, means pneumonia like organisms. Right? Pneumonia like organisms. Secondly, they are called as jokers of plant kingdom. Because they are changing their shape. MLOs, molecule like organisms. Molecule means soft skinned. Their covering is soft. As they don't have a cell wall, they are soft skin, means the plasma membrane is surrounding them. They can change their shape because the rigid wall is absent around them. Pleomorphic or pleuromorphic. Pleomorphic or pleuromorphic type of examples. So these are the things that we are discussing additionally about Mycoplasma. And as I said to you, that there is something which is more relevant. Completely lack a cell wall, because there are some examples. Because they completely lack cell wall, we can call them as CWD, cell wall deficient. Now there are the examples which lack cell wall, but when feed it properly, when get all the type of nutrition, they will again form the cell wall around them. One such example is L-form bacteria. The L-form bacteria is made specially to understand that how the cell wall is formed. Okay, so here is the thing that we are going to mention separately about the Mycoplasma. Remember that one. Here, cell wall deficient L-form bacteria is another example. But L-form bacteria can achieve the cell wall. Remember, other CWD or cell wall deficient example is Mycoplasma. Okay, here is the thing, they are smallest living cell. I'll tell you how they are smallest one. Mycoplasmas are considered as what? They are exception to monerans, exception to kingdom Monera. In two terms. First term, they can pass through the bacterial filter. If they can pass the bacterial filter, the second character is they can pass the bacterial filter. One thing. Regarding this, they are behaving as virus. They are becoming like virus. Therefore, mycoplasmas or PPLO or MLO are connecting link between bacteria and virus. They are connecting link between bacteria and virus. Number secondly, if you used to discuss the second reason why they are exception to kingdom Monera, because they lack cell wall. Okay, they lack cell wall. Remember this thing. So we need to relate. Okay, we need to relate these things very clearly. Number second analysis from this point. If they are passing the bacterial filter, it means they are smaller than bacteria, because bacterial filter is trapping bacteria. Smaller than other monerans, obviously. This is how it is established that they are the smallest one. Okay, so that type of character we used to study about Mycoplasma. Now, if I need to compare Mycoplasma with bacterial cells, I'll say something are different. What? Those things, both Mycoplasma and bacteria are prokaryotes, but Mycoplasma reproduce. We are saying that in the medium, Mycoplasmas in medium, they form fried egg-like colonies. Okay, they are forming fried egg-like colonies by the method which is called as elementary bodies. So they reproduce by some other method. They are not showing binary fission. Remember, in which terms they are different from bacteria? One, they pass through the bacterial filter. Number two, they are forming elementary bodies, right? Number third, they are different from rest of the Eubacteria because they never have a cell wall around them. So this is about the Mycoplasma and the kingdom Monera. Here we are going to conclude. Be clear about one thing, that you are supposed to attempt the daily practice paper just after the class so that your retention can increase. It hardly takes 10 minutes to answer those 15 questions from the classroom program. So it's very easy one. It's not at all much time taking. Those who are watching us on other devices like television or on the YouTube, for them it is better to download the Yupp Master app so that they can watch the previous videos, they can have the test papers, they can have the practice papers on the app only, okay, not on the smart TVs. Thank you for watching and all the best, my dear future medicos.